Thank you, Thomas. So I'm going to briefly present the M3P platforms where I work, and then Thomas asked me to talk about which is the strategy that we follow on environmental characterization. And then I will show some examples of how we use this environmental information for trait dissection. So M3P stands for Montpellier Plant Phenotyping Platforms and holds 3 different platforms. The first one is Phenopsis, that is devoted to the analysis of plant plasticity in Arabidopsis thaliana and response to different stress. Phenodin is a platform for measuring organ elongation rate and plant transpiration with a high resolution, typically from minutes, and is focused especially on leaf elongation rate, and it responds to drought and other environmental cues. And finally, PhenoArch, that is a platform for analyzing the genetic determinism of different plant responses to environmental conditions, mainly drought, light, and temperature, in a big panels, because it has a capacity for more than 2,000 plants. The common link of all these platforms is that we measure the plant response to environmental conditions. Okay, now most components of growth that we capture in those phenotyping platforms respond to local climate, and we are talking about how different processes respond to temperature, to light, CO2, etc. It's quite trivial, and also that what we found typically in these platforms, even in greenhouse or growth chambers, is that there's a large spatial and temporal variability of environmental conditions. And this has a consequence for phenotyping, and I would like to use this slide from the EPPN 2020 project, where we have a work package on environmental characterization, and these 4 points for me are essential. The first point states that we are not focusing in measuring just final biomass, we are capturing growth all over the cycle, so capturing the instantaneous environmental information is crucial if we want to give value to our phenotypic data. If we use, the second point is that we can use this instantaneous information for performing plant response curves to these environmental conditions. Third point is that we can use this accurate climate information for enabling the combination of data from different experiments, different platforms, and from platform to the field. And the fourth point is that climate information is a core component of modeling, as it drives most of growth processes. So the strategy that we have followed, or at least the things we do in our platforms, is as minimum having several sensors in each installation, and this is useful for compensating for the position of the sensor and having an idea of which is the spatial variability. Data has to be continuous logged, not only measuring minimum or maximum temperature, so we need measurements in a regular time lapse. And the environmental data has to be stored in a phenotyping, in a database or information system devoted to each platform. And when possible, having local environmental conditions as sensed by individual plants. And this can be achieved by placing the sensors directly to each plant, but this is not feasible when we have hundreds of plants. And or having precise maps of light or temperature or other environmental variables. And I'm going to show just quickly a few of the methods that we used. This was presented in the previous IPPS in Mexico. This is a method for mapping the local light in a greenhouse by using hemispherical images. So we can take hemispherical images, we can simulate the sun paths for every hour, every day of the year, and then we can calculate how this maps for every day of the year. Similarly, we can do the same for temperature, but in this case, what we have used is a network of sensors. So we have placed 60 sensors placed in pods, and what we can obtain with this kind of wireless sensors is a map of temperature. Here you can see how important it is to measure the temperature in an instant basis, because it changes a lot. We can see shifts of temperature of more than 5 degrees. And depending on the rate that we are measuring, this can have a big consequence. And finally, this information has to be stored in a database, and this is what we do, but also, and not only the data, we store also the sensor characteristics, so which variable we are measuring, which are the units that are used for this variable, and also the events, when this sensor has been installed, calibrated, if this sensor has a dysfunction, etc. And of course, where the plants and sensors are at each time. And for more details, I will be presenting our information system on Thursday. So welcome if you want to see more in detail. So now I will talk how we use this information with some modeling for extracting relevant traits that can be used in genetics or in modeling. So if we take this, the equation of Monteith, where biomass can be explained as the amount of incident light, the amount of this light that is intercepted, and how this intercepted light is converted to biomass. This is one example that across the scales, and we can even dissect these terms of the equation, at least the genetic terms of this equation, in more simple and heritable traits. And for example, intercepted light that depends on the leaf area. We can say that leaf area also depends on the architecture and the canopy size, and the canopy size depends on the number of leaves, how big are those leaves, which is the rate of growth of these leaves, and which is the sensitivity of these leaves to the environment. And similarly, radiation use efficiency that is mainly driven by gas exchange, we can define it as other terms. And I will show now how some of the work that the group of Francois has done and on different traits, for example, plant architecture, leaf size, or sensitivity. In the case of sensitivity of leaf elongation rate to environmental conditions, this is mainly done in the platform phenotype, where we can combine leaf elongation rate measurements performed in the greenhouse, where, I don't know where is the pointer. Well, we can combine measurements in the platform, where plants experience fluctuation in environmental conditions, measurements in the chamber, where we measure stable control conditions, and also measurements in the field. And then we can combine all this information at different scales and with different scales to extract common genotypic responses to environmental conditions. In this case, is the response of leaf elongation rate to solve water potential or evaporative demand. And this response the response is highly heritable and can be used later in different models. Similarly, we go back to the equation of Monteith. This is the work of Sugei, and he used a 3D reconstruction plus a functional structural plan model combined with the local light maps that I showed before to estimate light interception and radiation use efficiency in 250 maize hybrids and different experiments. And he could dissect the genetic architecture of these traits and found interesting information. Similarly, this is the work of Santiago Alvarez Prado. He did some work on developing a method for estimating stomatal conductance based on transpiration, leaf area, and micrometeorological conditions. Basically, what he did, he inversed the Penman-Monteith equation. So with this information of transpiration, leaf area, and micrometeorological conditions, he estimated the stomatal conductance in 750 plants. And he found high genetic variability and heritability for this trait. And then he did a genetic dissection, and he built a model for predicting these stomatal conductance by using the detected QTLs plus the local microenvironmental conditions. I shift to another crop now, to grapevine. And this is nice work of Odd, that she was working with night transpiration, and she found that night transpiration is under tight genetic control in grapevine and strongly contributes to variation in water use efficiency. In this work, she combined the measurements done in PhenoArch at the whole plant level. She measured water use efficiency under fluctuation conditions, Then she moved the plants to a growth chamber with control conditions to have another range of conditions where she measured in a very specific condition the night and day transpiration. And she validated also these measurements in the field, where she also measured night and day transpiration, and she combined all the information to evaluate the genetic architecture of night transpiration, and she found common QTLs with water use efficiency. And this work has been published in PNAS. Similarly, this is a work of Emily. Thomas showed in a previous paper of Emily. And here she used the leaf appearance measurements that we perform usually in the platform, that are quite easy to perform in the platform but difficult to perform in the field, and that indeed they are very high heritable. So she used these leaf appearance rates that are very heritable, and she used this information from the platform to establish in the field, in the same genotypes in a network of fields, phenology and different critical window periods where plants may experience different scenarios of drought, high temperature, or other stresses. And she used this sensibility to the environment with the genetic information to build a model, and she performed a genetic prediction. So basically these are, I went a bit fast, but these are some examples that we use in Montpellier to combine this environmental information that we aim to characterize in a high precise way with different types of modeling to extract traits that can be used in genetic analysis. So as a take-home message, I would like to put again these 4 points of the EPPM project that for me are quite important. So, thank you.